Good day, everybody. This is Chris back again at the Ancient Scholar, and today what I'd like to talk about is the liver. Specifically, I'd like to talk about the histology of the liver. And when we look at the liver and we actually look at the microscopic structure of the liver, we can organize the liver in a few different ways. And what I want to do is I just want to talk about generally what we'd see if we were to take a slice of tissue from the liver and take a look at it. We would see something that looks sort of like the picture here. We'd have these kind of hexagonal shaped units that kind of fit together in almost a honeycomb-like shape. And within these units, we'll talk about some of the major structures. So some of the major structures that we have within these units, you want to call them that, are the actual hepatic cells, the functional cells, if you want to look at it that way, and they're all organized inside of these honeycomb structures. So I'm just going to kind of draw all these cells, what we call hepatocytes. All right, so you have lots of hepatic cells, different types of cells doing different things. You have immune cells as well, lymphatics, etc. So you have all of the functional cells of the liver, the hepatocytes, so we'll just collectively call them, inside of this five-sided structure here. Okay, so what else do we have? Well, what you have is on each side of the structure here, okay, at each corner of the structure, you have a group of three different structures. So I'm going to draw those little guys in there. All right, here we go. And they're everywhere, at every side here. All right, so just like that. A group of three different structures. Okay, those little guys there. And those are what we call the portal triads. The portal triad. All right, and you have one basically at every little corner of these structures. Okay, so you have the hepatocytes here in the middle, you have the portal triads, and then in the middle of these structures, you also have another structure, and let me just draw that in a different color, and that is a central vein. All right, so these are the major components. You have your hepatocytes packed in here inside of these structures. In the middle you have a central vein, and then on each corner you have the portal triad. Now the portal triad, it contains three different structures, and we'll talk about them in a little more detail as we go along. Okay, so they contain a portal vein. Okay, they contain a hepatic artery. All right, and they also contain a bile duct. So this is kind of interesting. So the hepatic artery, as you might imagine, brings oxygenated blood into the liver. But the portal vein is interesting in that it's bringing blood into the liver, but it is not oxygenated blood. So where is that blood coming from? Well, I mean oxygenated, I mean it's not arterial blood. So the portal vein is actually blood that comes from the gastrointestinal tract. Okay, and this is of course very important when we talk about the concept of first pass metabolism. So what happens is nutrients, drugs, toxins, what have you, are absorbed through the alimentary canal, and then they go into the portal circulation, okay, which ultimately goes to the liver. Okay, this is venous blood, and then from the liver, okay, so from the portal vein, these substances will work their way through these structures into the portal vein, and finally make their way to the inferior vena cava, where they enter the central venous circulation, go to the heart, and then are distributed through the heart, the lungs, and then out into the arterial system. Okay, so this is kind of an interesting and a unique thing that happens here. That you actually have venous circulation coming into the liver. And that's important because the liver is so metabolically active that we want to metabolize stuff that is being absorbed through the GI tract. So instead of going from the GI tract to the heart, and then the heart circulates it and it eventually makes its way to the liver through evolution, the circulation is designed, or not designed, evolved rather, uh, to go from the GI tract via the portal circulation right into the liver. Um, so it can be metabolized very quickly and very early on following absorption. Okay, so what I'm going to do now is I'm going to look at this architecture, if you want to call it that, of the liver, and I'm going to look at it in a few different ways, and each way is going to focus it is going to make one or more of these components more central to the picture. So we're gonna look at three different pictures of the the primary architecture of the liver, and the three pictures include the following. One is the lobule picture, which is a classical picture. The second is the portal lobule picture. And then the third, which is going to be the most important when considering toxicology and pharmacology, is the hepatic acinus picture. Hepatic acinus. All right. Now, these are all just three different ways of looking at this picture here. Okay, so just kind of keep that in mind as we move on. So let's go ahead and look at the lobule, or the classical lobule picture first. Okay, so the classical lobule picture takes one of those units and isolates it into a structure called the lobule. So here we have the lobule, or lobule, I call it lobule. And so what we have here is we have the triads. Okay, we're looking at the triads, and then of course we're looking here at the central vein. All right, this picture is nice because it can kind of give us a good qualitative idea of where stuff is flowing through this lobule, through this structure here. So let's talk about how stuff is flowing. So what's going to happen? Well, we should have a good idea of how arterial and venous flow. So when you look at perfusing tissues, you have an artery here, right? You have an artery, and you have oxygenated blood diffuses through the tissues. Okay, so oxygenated blood comes into the tissues through an artery, and then it leaves the tissues through a vein. All right, deoxygenated blood leaves the tissues through the vein. And that is a pretty intuitive picture, and that is a picture that is also happening here in the liver. So this little blue structure here, we'll just go ahead and label our structures real quick. This little blue structure here is the portal vein. Okay, this red structure here is the hepatic artery, and then this little green structure here is the bile duct. All right, so let's see what's going to happen here. So what's going to happen is blood is coming into this lobule through the hepatic artery, and then that blood can perfuse these hepatocytes. So the arterial to venous circulation is going to move like this. Blood is going to start here, and it is going to move through, perfuse hepatocytes, and then that arterial blood is then going to be taken away from the liver through the central vein. Okay, that's pretty intuitive, not a big deal there. I think that makes a lot of sense. So you have this going on here. All right, fair enough. Okay, Well, what else is going on? Well, here's an interesting thing. Blood that comes into the liver through the portal vein, okay? So blood coming in through a vein is also, through the portal vein, will also move toward the central vein. So not only do I have oxygenated blood moving, but I have this kind of this deoxygenated blood that's moving from the portal veins toward the central vein, all right? And that's important because that blood is coming from the intestines, so that blood may have nutrients, it may have drugs and toxins and substances that need to be metabolized. So as that venous blood, that portal venous blood is moving through this lobule um, or lobule, it is being metabolized, and then once I have all of my metabolites that are created, those will then go into the central vein, and then they will leave the liver, go into the inferior vena cava, go through the heart, to the lungs, brain, out to the body, etc. All right. And then the one thing that runs opposite to this from the triad to the central vein, at this point everything is running from the triad to the central vein, the opposite is occurring when it comes to bile. Bile is basically moving from the hepatocytes toward the bile duct. Okay, so bile is moving in the opposite direction if you want to think of it that way. That's probably a decent model. Okay, so bile is moving in this direction from the central vein toward the bile ducts. So this view, this classical lobule view, is good at it. It kind of gives us a good overall look at how things are going. Okay, so now what I'm going to do is I am going to look at the portal lobule view. Okay, so that was a classical. Now what I'm going to do is I'm going to look at the portal lobule view. And with the portal lobule view, what I do is I look at a few of these hepatic lobules, and what I do is I just kind of draw an arbitrary triangle. It's not so arbitrary, but I'm going to make a triangle. What I'm going to do is I am going to start here, okay? Remember, this is a central vein, and I'm going to connect these three central veins, these three units here, okay? So I'm just going to make a triangle here, and I'm going to call this my portal lobule. This is not this view here, which is a classical lobule, where I'm just looking at one of these, but the portal lobules, I'm looking at three of these, and I'm drawing a triangle. Okay, I'm drawing a triangle that connects three of the central veins. And then remember what we have. Remember we have these little triads here, and I have, where these three come together, I have a triad right in the middle here. And so what this view, this way of looking at the architecture of the liver is nice in that we can focus on what's going on primarily with what's coming into the triad. So the portal lobule is really making the triad the kind of the central structure, whereas the classical lobule view kind of makes the central vein the central structure. Here we're actually focusing on the triad, and so this is really nice because what we can do is this is a cool way of basically drawing lines from these portal from the central veins toward the triad, and the only thing that happens, the only thing that moves that way is the bile. So the portal lobule view is a good way of focusing on how the bile moves. The bile is moving toward the triad, toward the bile duct in that triad. Okay, and now probably the most important view in terms of toxicology and pharmacology is what's known as the acinus view. The acinus view. Okay, so the hepatic acinus. All right, and what I'm going to do here, so here I have my my lobules I have two lobules that are kind of next to each other. I have the central vein, and then I have my triads here. And what I'm going to do is I'm going to kind of draw a diamond shape. Let's make that a different color. So I'm going to start at my central vein, and I'm going to draw it through here, and then through here, and then I'm going to do the same on this side. So I kind of have a diamond shape here, okay? And this is really nice because this really is the view that helps us conceptualize the perfusion, okay? The perfusion through the architecture of the liver. So what can we infer from this picture here? Well, what we can infer is that the tissue, the hepatocytes that are closest to the triads are going to be well perfused. They're closest to the arterial blood supply. So this area, okay, in here within that triangle that I drew, and maybe I'll put some red in here. This area here is going to be highly perfused. It's going to receive high amounts of oxygenated blood. And so that area that's directly kind of in between these two triads here, that's well perfused, we call zone 1 of the acinus. Okay, zone 1 of the acinus. All right. This zone 1 is also known as the central lobular zone, or the periportal zone, okay, or zone 1. What do we want to know about zone 1? Well, the big thing to know about zone 1 is that it is highly perfused. Okay, so I have high levels of oxygen in this zone. And just kind of as an aside, this zone is particularly susceptible to viral hepatitis. All right, so in patients that have viral hepatitis, this area here is often the area attacked. And when I have states of ischemic states where I have compromised perfusion to the liver, so I have compromised perfusion to the liver, these areas tend to be less vulnerable to ischemia because they will receive more oxygen than the other parts of the acinus in a shocky or a low perfusion state. Okay, so that's zone 1. So then what we do is we have kind of this intermediate zone here, out here, that's kind of in the middle. So I'll just draw that in maybe a different color here. All right, so this zone here. And this zone here is further away, but not furthest away. It's kind of in between, and this is what we call the zone 2. Okay, this is zone 2 of the liver. It's kind of an intermediate zone. Not a whole lot to say about that in terms of toxicology. I believe that, what is it they say? That this zone is more vulnerable to like yellow fever. People that get yellow fever, the hepatocytes in this zone tend to be more vulnerable. But again, that's not something that I've studied in detail, so I'll be kind of careful about making definitive statements there. But zone 3 is going to be really, really important when it comes to toxicology and pharmacology, and that is this zone out here. This zone out here, okay, which is what we call zone 3. Zone 3, it is also known as the pericentral zone. The pericentral zone. And this is the zone where we have a significant number of cytochrome P450 enzymes. All right, this is where I have a significant number of cytochrome P450 enzymes working. So this area here is sometimes known as the detox center. The detox center of the liver. Okay, so in this area here in zone 3, I am going to have lots of metabolites being produced. And some of these metabolites, of course, are going to be toxic. And in particular, what we run into in particular is we have the highest number of CYP2E1 enzymes. And if you guys have been watching my videos on some of the toxic substances, you will know that CYP2E1 enzymes are particularly important when it comes to the metabolism of acetaminophen to its toxic metabolite NAPQI. So this zone is going to be particularly at risk for acetaminophen toxicity, because this is where you're going to have higher levels of the NAPQI metabolite, and so you're going to have a much higher risk for hepatotoxicity. In addition to that, this zone is much more sensitive to ischemic changes. Because this zone is already, this zone is already relatively hypoxic compared to the other two zones. So in low flow or ischemic states, zone 3 is going to be at higher risk. Before I go ahead and end this video, just a couple of things that you might want to know about zone 1. Because it is oxygen rich, you would expect a lot of oxidation to occur here, lots of oxidation. So processes such as gluconeogenesis, beta oxidation of fatty acids will predominantly occur here, and even the creation or making of cholesterol will be occurring. So a lot of oxidative processes. However, zone 3 is where it's at when it comes to your cytochrome P450 enzymes. Okay, guys, I think I'm going to go ahead and cut it off here. Hopefully that made some sense, and hopefully you enjoyed that. As always, thanks for hanging in there.